Neanderthals were not replaced. They became modernized. Having big differences in skull and skeletal morphology doesn't mean there is a big difference in genetics. It only takes a few specific genes to make those changes. When Neanderthal genomes were first fully sequenced, the striking result was that more than 99.7% of genetic base pairs are identical to those of present-day humans, meaning only about 0.3% differ. Crucially, Neanderthals did not possess a fundamentally different set of genes from modern humans. Rather, they carried slightly different versions of largely the same genes, many of which were later absorbed into modern Eurasian populations through introgression. In genetic terms, Neanderthals and modern humans are therefore not separated by different genomic architectures, but only by a thin layer of variation, reinforcing the view that their distinction is one of population divergence, not biological incompatibility or true species separation. But instead of emphasizing this similarity, the mainstream narrative emphasized differences and told the public that interbreeding was limited to a single event 50,000 years ago. Nothing could be further from the truth. Neanderthals were not genetically distant primitives, but a closely allied European population exhibiting subtle anatomical and regulatory differences, rather than a fundamentally separate human species. And the distinction between Neanderthal and modern Homo sapiens genomes is therefore one of emphasis and expression, not structural revolution. The Neanderthal genome also shows a remarkable continuity of pigmentation and phenotypes between Neanderthals and modern Europeans. When a Neanderthal genome is compared to a modern human genome, the measured difference amounts to approximately 0.12 to 0.3% of base pairs, a figure only marginally greater than the variation observed between two random modern humans who differ at roughly 3 to 5 million base pairs as well, meaning that the Neanderthal-modern human gap is only slightly larger than normal human-to-human variation. Out of the approximately 3.2 billion base pairs that compose the human genome, only 4 million differ in some way, and only about 100,000 of these represent consistent lineage divergence, with just 300 to 400 variants connected to detectable behavioral, physiological, or cognitive differences, a scale of divergence that is extraordinarily small for two populations once confidently labelled as separate species, and that instead supports the view that they were deeply compatible, interbreeding human populations separated by modest evolutionary drift rather than true biological discontinuity. For over 40 years, the dominant story told to the public has been simple, dramatic, and emotionally satisfying. Anatomically modern humans emerged in Africa, swept outward like a tidal wave, and replaced the archaic Neanderthals of Europe and Western Asia. This narrative has been repeated so often that it has hardened into cultural fact. Yet, the genomic revolution has dismantled the very foundations of this story. What the data now reveal is not a tale of conquest and extinction, but one of continuity, assimilation, and transformation. The more deeply we look into the Neanderthal genome, the more evident it becomes that Neanderthals did not vanish as a separate species, but were gradually folded into the population we now call Homo sapiens. In other words, there was no true replacement. There was evolution through integration, and the nail in the coffin to simplistic narratives of replacement. When the first Neanderthal genomes were sequenced in 2010, the results shocked a discipline long trained to emphasize difference over similarity. Europeans and Asians were shown to carry between 1 and 4% Neanderthal DNA, a finding that immediately contradicted the idea of a clean break or total displacement. But even this percentage understates the deeper reality. The most commonly cited data only count easily identifiable Neanderthal segments, not the shared ancestral structures and deeply conserved genomic architecture that blur the boundary between what has been labelled Neanderthal and modern human. Across the entire genome, Neanderthals and Homo sapiens are approximately 99.7% identical. This degree of similarity is not the signature of separate species locked in evolutionary isolation. It is the signature of populations that diverged, drifted, and then reconverged through sustained interbreeding. What this means is that Europeans are the direct descendants of Neanderthals, not recent arrivals. It means Eurasians can claim 300,000 years of continuity in Europe and Asia. The very concept of replacement collapses under this genetic closeness. Replacement implies that one population disappears and is superseded by another, as wolves might be replaced by domesticated dogs or one language extinguished by another. But the genetic reality is one of flow, rather than erasure. Neanderthal alleles are not relics trapped in a few isolated individuals. They are distributed throughout Eurasian populations, embedded in genes related to skin pigmentation, immune response, keratin production, fat metabolism, and altitude adaptation. These contributions are not trivial. They are functional, selected, and persist because they were beneficial. A population that is replaced does not leave behind a biologically active legacy across vast regions and tens of thousands of years. It leaves absence. Neanderthals left continuity. The replacement model also assumes that modern humans were a fully formed, genetically coherent entity arriving from Africa to erase what came before. This is a myth. Early Homo sapiens populations were themselves heterogeneous, regionally structured, and far from a single uniform type. Africa was not a factory producing a standardized model of humanity. It was, and remains, a landscape of deep genetic subdivision. Populations that would later be labeled modern humans already exhibited enormous variation before any encounter with Neanderthals. When such populations moved into Eurasia, they did not meet a wholly alien species. They encountered close evolutionary cousins who had adapted to those lands while retaining profound genetic overlap. The evidence for continuity extends far beyond simple percentages of introgressed DNA. Neanderthal-derived genes participate in core biological systems of living Eurasians. Immune genes, such as those associated with toll-like receptors, derive partly from Neanderthal ancestry, enhancing response to Eurasian pathogens. Skin and hair genes show Neanderthal-associated variants linked to cold adaptation and UV response. Even aspects of brain development appear influenced by Neanderthal genomic contribution. These are not pale echoes of a vanquished people. These are structural components of what modern Europeans are. If Neanderthals were truly replaced, then natural selection would have purged their genetic material entirely. Instead, selection preserved and amplified it. This preservation is not accidental. It reflects biological compatibility and repeated gene flow. Replacement models require not only extinction, but genetic sterilization. What happened instead was absorption. Morphology, too, tells a story of convergence rather than rupture. The fossil record, when examined without ideological filtering, reveals transitional traits in so-called modern humans that echo classic Neanderthal anatomy. Occipital buns, supraorbital robustness, midfacial projection and dental patterning persist into early Upper Paleolithic Europeans. These features do not vanish overnight. They fade gradually, as one would expect from populations blending over time. Evolution does not operate like political revolution, it operates like sediment deposition, layering and intermingling until boundaries become diffuse. The perception that Neanderthals were fundamentally different was cultivated in an era before genetics could speak. Early reconstructions caricatured them as brutish, bent-kneed creatures, closer to apes than to ourselves. These images were not scientific conclusions. They were cultural projections shaped by social hierarchy and ideology. Modern DNA sequencing has undone this fiction. The Neanderthal genome demonstrates that the difference between them and us is smaller than the difference between many modern human groups. Consider this carefully. The genetic distance between Neanderthals and present-day Eurasians is comparable to, or even less than, the distance separating deeply diverged African populations from one another. If genetic distance does not justify separation within Africa, it cannot justify it between Neanderthals and Eurasians either. The insistence on Neanderthal otherness thus becomes ideological, not scientific. The theory of replacement also relies heavily on a false assumption of cultural superiority. It presumes that modern humans possess symbolic thought, language, and complex behavior, whereas Neanderthals did not. This assumption has been steadily eroded. Neanderthals buried their dead, used pigments, created composite tools, exploited marine resources, controlled fire, and likely engaged in ritual behavior. There is no evidence of a civilizational gulf requiring replacement. Instead, we see two closely related populations meeting, interacting, and merging. Archaeological layers that were once interpreted as abrupt population turnovers now look more like periods of cultural transition and mixing. Tool traditions show overlap and gradual modification, not sudden disappearance. Sites across Europe reveal continuities that are incompatible with wholesale extinction. Where population replacement did occur, it was not one-sided. It was mutual and transformative. The genome thus reframes the entire story. Rather than modern humans replacing Neanderthals, modern humans are the product of Neanderthal integration combined with modern human ancestry. The term Neanderthal itself becomes a historical label rather than a biological boundary. They were not a separate evolutionary path that failed. They were part of the braided stream that produced contemporary humanity.
Interesting question in bringing a Neanderthal to life is skin colour. Now this would probably have varied Neanderthal to Neanderthal, but DNA samples of Neanderthal specimens from modern Europe found that those individuals probably had pale skin and some possibly even red hair. And lighter skin means that you can produce more vitamin D even if you have less sunlight. So essentially, before Neanderthals, there was no need for lighter skin, and that's why we didn't have white people until then, because they hadn't been invented yet.
The idea that Neanderthals, who are hyper-adapted to the cold climates of Eurasia, retained an ancestral pigmentation type is so biologically improbable, it is totally absurd. To insist on replacement is to cling to a 19th century vision of evolution that sees history as a ladder, where the superior overwrites the inferior. Modern genetics reveals a network, not a hierarchy. Neanderthals were not evolutionary dead ends. They were contributors, ancestors, and foundations. Their disappearance as a distinct population was not extinction in the sense of total loss, but transformation through integration. The data no longer permit the language of replacement except as metaphor. Scientifically, it is inaccurate. There was no moment when modern humans arrived and wiped clean the Neanderthal slate. Instead, there were generations of proximity, intimacy, reproduction, and shared survival that dissolved artificial boundaries. The Neanderthal end was not a fall, it was a merger. This conclusion is reinforced by increasingly sophisticated population genetic modeling. Analyses based on D statistics consistently demonstrate asymmetric gene flow from Neanderthals into modern populations, but also reveal evidence for multiple pulses of admixture rather than a single, brief event. Coalescent simulations indicate that Neanderthals and Homo sapiens diverged roughly 500,000 to 600,000 years ago, but maintained intermittent connectivity, with effective population sizes that fluctuated and occasionally overlapped. Such divergence times, when paired with extensive later introgression, describe populations that separated but never achieved complete reproductive isolation. The length and distribution of Neanderthal-derived haplotypes further underscore this point. Early modern Eurasians carried long, contiguous Neanderthal segments, which over tens of thousands of years were broken down by recombination into shorter tracts. The persistence and measurable decay of these tracts across contemporary populations reveal a long-term assimilation process rather than a short-lived hybridization followed by extinction. Linkage disequilibrium patterns show selective retention of certain Neanderthal variants while purging others, demonstrating active evolutionary integration rather than passive fossilization of archaic DNA. Even claims of strong negative selection against Neanderthal DNA reinforce the assimilation model rather than contradict it. Selection does not operate unless hybridization has occurred. The very need to purge incompatible variants presupposes extensive gene flow and genomic mixing. Over time, what remained were variants that harmonized with the Homo sapiens genomic background, becoming indistinguishable from so-called modern alleles in their functional contribution. The genome thus reframes the entire story. Rather than modern humans replacing Neanderthals, modern humans are the product of Neanderthal integration combined with African and Eurasian ancestry. The term Neanderthal itself becomes a historical label rather than a biological boundary. They were not a separate evolutionary path that failed. They were part of the braided stream that produced contemporary humanity. This view does not diminish Africa's importance. It enriches it. Africa remains the deepest reservoir of human genetic diversity, but diversity does not imply isolation. The human story is not one of purity and replacement, but of constant contact, migration, and recombination. Eurasian populations today carry African ancestry, Neanderthal ancestry, and ancient Eurasian ancestry in varying proportions. All of these streams converge in what we call modern humans. What vanished was a label, not a lineage. They persist in our bones, our immune systems, our physiology, and our very DNA. Their story is not extinction. Their story is us. But don't believe the narrative that anthropology never predicted, that Neanderthals and modern humans had interbred. This was actually a popular theory as long as 100 years ago, but it was buried in a wave of National Geographic headlines of pure humans evolving in isolated rift valleys and then spontaneously emerging to conquer the world. The most compelling evidence that Neanderthals were integrated rather than replaced lies in the functional impact of their introgressed genes. These are not random genetic scraps, but biologically meaningful variants that altered phenotype and improved survival in European environments. Skin and pigmentation-related genes show equally significant introgression. Multiple Neanderthal-derived variants occur in genes such as BNC2, contributing to differences in hair and skin pigmentation and response to ultraviolet radiation in 70% of Europeans. These variants facilitated adaptation to lower UV environments and colder climates by altering barrier function and pigmentation regulation. Many other Neanderthals' genes also influence eye, skin, and hair pigmentation, reflecting their deep integration into the architecture of modern Eurasian skin biology. These adaptations highlight physiological compatibility and the selective advantage conferred by Neanderthal genetics in Ice Age Eurasia. Neurodevelopmental and circadian rhythm genes also show introgress contributions. Certain Neanderthal haplotypes influence expression timing in clock-related genes, affecting sleep cycles and seasonal physiology, again demonstrating deep biological integration into complex regulatory systems. Such involvement in core regulatory networks would not be possible had Neanderthals been simply replaced. Only populations that merged and continued evolving together could produce this level of functional genomic entanglement. Population genetic modeling provides quantitative context for this continuity. Whole genome comparisons indicate that the lineages ancestral to Neanderthals and Homo sapiens diverged approximately 500,000 to 600,000 years ago, a time frame consistent across multiple models using mutation rates calibrated through fossil and ancient DNA benchmarks. This divergence, however, did not mark complete reproductive isolation. Instead, analyses show long periods of partial connectivity, with demographic bottlenecks and expansions shaping both lineages in parallel. Effective population size estimates reveal that Neanderthal populations were small and fragmented compared to the larger, more subdivided Homo sapiens populations. Yet despite this, gene flow persisted. Simulations indicate that even low levels of recurrent interbreeding over thousands of years are sufficient to produce the introgression patterns observed today. These models support scenarios of repeated contact episodes rather than a single admixture pulse. Importantly, the genetic divergence between Neanderthals and modern humans, measured by average nucleotide differences and pairwise coalescent times, overlaps with divergence between some extant modern populations. This overlap weakens any justification for defining Neanderthals as a fully separate species and strengthens the argument for viewing them as a regional structural variant within the broader Homo sapiens continuum. Genetic drift, selection, and recombination shaped the final pattern. Neanderthal-derived segments were initially widespread after admixture but gradually filtered through purifying selection leaving behind alleles compatible with the sapiens' genomic background. This gradual genomic winnowing is exactly what one would expect from a long-term assimilation process, not a replacement event. Together, functional introgression and demographic modeling converge on the same conclusion. Modern Eurasians did not arise from the extermination of Neanderthals, but from their genetic, biological, and evolutionary incorporation. The Neanderthal genome did not die. It dissolved into the expanding human population, altering its structure, enriching its diversity, and shaping its adaptive potential for tens of thousands of years to come. The assimilation model described here is not speculative. It is built upon a substantial body of peer-reviewed genetic work produced over the last 15 years by many of the most respected researchers in paleogenomics and evolutionary biology. The foundational breakthrough came from the work of Svante Pääbo and his team at the Max Planck Institute for Evolutionary Anthropology. The 2010 draft sequence, published by Richard Green, Johannes Krauss and colleagues, demonstrated conclusively that almost all modern humans share a measurable proportion of Neanderthal ancestry, overturning the long-standing assumption of total replacement. This work established that Neanderthals and Homo sapiens interbred, and that Neanderthal DNA is present in all populations today. Subsequent refinements produced high-coverage Neanderthal genomes from Vindiger and Altai specimens. These studies confirmed multiple pulses of admixture and demonstrated that different modern populations carry different Neanderthal source contributions, further undermining any idea of a single replacement episode. Sriram Sankararaman and David Reich made critical contributions through their work on local ancestry detection and introgression mapping. Their analyses identified Neanderthal-derived segments across the modern human genome and demonstrated that these segments cluster in genes associated with immunity, pigmentation, and environmental adaptation. Reich's later synthesis explicitly argued against strict replacement models and emphasized the mosaic nature of early human populations. Joshua Akey further expanded this work by showing that Neanderthal introgression was not uniform but varied by region and population, revealing selective preservation of specific Neanderthal variants. Their studies highlighted that introgression was both deep and functionally significant, not transient or marginal. Their work demonstrated that Neanderthal DNA was not merely tolerated, but actively favored in many Eurasian environments. Others working on fine-scale population genomic modeling demonstrated the complexity of admixture timing and the persistence of Neanderthal haplotypes across millennia, evidence inconsistent with a model of brief contact followed by extinction. Critically, the integration of these genomic findings into paleoanthropological interpretation has been championed by scientists such as J. Bertrand Petit, Josh Akey, and David Reich, all of whom have acknowledged that the classical replacement framework no longer reflects the genetic evidence. Even these top scientists who once promoted the simple replacement narrative have to speak in veiled language while they slowly chip away at the old theory using terms such as extra-African origins. Even those who once defended strict replacement models now recognize that the reality was more intricate, involving repeated contact, gene flow, and regional continuity. Together, these studies and researchers converge on a single empirical truth. Neanderthals were not erased. They became us. Their genomes merged with those of early Homo sapiens, producing the populations that would spread across Eurasia. Replacement did not occur. Transformation did. Thanks for watching.